Welcome to our Bioscience strand. I'm here with Mr. Kessler, Eric Kessler. He's our strand leader in Bioscience. Mr. Kessler, can you kind of show us some of the great products and processes that you have in this area?
Sure. First, this classroom is one of the teaching labs for the Machinery of Life class here that we have in the Bioscience strand at the Blue Valley Center for Advanced Professional Studies. We have a number of industry standard machinery in the classroom. We have a chemical fume hood that you have in the back there. We have a biological safety cabinet for students working with strains of bacteria. We have a real-time PCR machine, standard centrifuges, shaking incubators, things like that you have in a standard laboratory that you would find, at the Stowers Institute or academic and other business institutions in the area. Okay. If you'd follow me, this is Mrs. Tehizada. She's the teacher of this class at the moment, and she's going to introduce you to some of the things her students are doing today.
Today we are doing a crime scene investigative lab, which has two components. It has a PCR component as well as a gel electrophoresis. And we did our PCR yesterday, and students today are actually loading up their gels. So if you come back here, I'll introduce you to three of our students who are actually right now in the process of getting ready to load their gels. This is Libby, Carrie, and Nazanin. Libby, can you please let us know what Nazanin is doing right now?
Yeah, right now Nazanin is putting our DNA that has been amplified in the PCR machine into her gel, which we poured earlier. This is a stained DNA, and we will run it in the electrophoresis and see how it comes out and compare it to the allele ladder. That will let us know which suspect is the person who committed the crime. So I'll pass it off to Mr. Kessler now.
Okay, so welcome back. This second room that we're going to be proceeding through is our research bioscience space for students that have taken the Machinery of Life course and want to study more in depth about some particular aspect of biology. This is Bert Gowan. He's going to tell you a little bit of some things he's been doing recently.
This right here is a protein modeling project. The software I'm using is called Rasmo, and what it can allow me to do is take protein files and create different models. This is a Shiga toxin, and I've been working with the B pentameter of the Shiga toxin, modeling that, and working with the amino acids on that. And once I finish up this model over here, I can send this file, this digital file, into this printer over here, and this printer can create actual 3D physical models like this, and I can use this as a model for other students and other projects.
So if you follow me, I'll talk a little bit about this room as we head to our final space. So the research room that you're in, it's a little bit different than the other labs, and it's not built really for teaching. This is more for students to actively work on independent research projects, and you can kind of see that in the space around you. As we move on, the last space here is for our Rhythms of Life class. In here, students look at multicellular organisms, both as model organisms and also in the field. And so here's the second of my colleagues, Joe Whalen, and he will tell you a little bit about what his students are doing today.
Well, today my students are examining some prairie grasses and prairie flowers that they have harvested last week at a local tallgrass prairie, and they're trying to identify the various species that are growing there to kind of get a bigger picture of what's been occurring there since it's been farmed ever, so many decades ago. One of you folks care to let the folks out there know what you're up to? Brad, Evan.
Okay, well, we're just identifying these plants using these field guides we have. Basically, what you're trying to do is just match up as many of the physical features of the plant to the book as you can. And this one appears to be goldenrod. And then we are going to be pressing these plants. So, Austin, would you like to demonstrate pressing this for us?
Getting it down to a preservable size.
Basically, for pressing this, the purpose of this is we're going to be getting them flat and then having them dry out so you can preserve them and use them, look at them at later times as a reference, or keep them if you need them for some type of research or something that you're doing. So what he's going to be doing right now is he's cutting it down into smaller sections so that it'll fit on the paper that we have for it. and then we will be pressing it. What he's doing now is cutting off all the excess vegetation that's not needed necessarily to identify it. And then, once he's done with that, he'll just lay it flat and cover it with a newspaper and then another piece of cardboard.
So can you tell me a little bit about what this program's done for you and opening your horizons?
For me, what this has done is it's really gotten me, like, it just is an eye-opener, really. It's allowed me to go in here and just see what's out there in the biological field for me. I'm very interested in it and just didn't really know what kind of options were available to me if I were to go into it, so this has been a nice opener.
Thank you so much for sharing with us, and thank you so much for your time. And I want to thank you as well, Caps, and I'm sure you're going to have a lot of questions, and I'm sure the people there are very well capable of answering them for you. So take care. Thank you.